To find the actual origin of Alzheimer's disease, we must look past the macrostructure of the brain, past the cortex, and travel deep into the fluid, abstract space inside a single neuron. For decades, the pathology of this disease has been narrated as a story of two misfolded proteins, amyloid beta plaques outside the cell and tau tangles within it. But clinical literature records two severe metabolic crises that appear long before those plaques ever deposit. First, the aging Alzheimer's brain accumulates high, toxic levels of ceramide, a tightly packed waxy sphingolipid. Second, the neuron suffers a bioenergetic collapse. The mitochondria fail, and cellular respiration falls. Researchers have spent 30 years treating the lipid accumulation and the energy failure as two distinct diseases occurring in the same brain. They are the exact same lesion. The lipid disturbance and energy failure are the manufacturing output and mechanical failure of one physical organelle contact. This 2D schematic illustrates that junction, the mitochondria-associated membrane, or MAM. It is where the endoplasmic reticulum and mitochondrion align. The defining physical parameter of the MAM is distance. The two organelles maintain a strictly controlled gap of 10 to 30 nanometers. The earliest stage of Alzheimer's begins right here, as a spatial disorder at a junction too small to stain. The primary rule of this junction is that it is a weld that never fuses. The two membranes must remain extremely close, but they must remain apart. To maintain this exact distance while the organelles move and divide, the cell bridges the gap with a physical scaffold of protein tethers. This buildup shows the calcium tether. On the ER sits the IP3R channel, linked by GRP75, directly to the VDAC channel on the mitochondrion. At baseline, it acts as a calibrated synapse, delivering rhythmic pulses of calcium across the gap. The ER side of the gap has its own specialized composition. Structurally, this localized zone is an intracellular liquid raft, heavily enriched in cholesterol. Crucially, this raft is the designated home for ceramide synthase enzymes, which lock directly into this domain. In lipid biology, there is a strict topology principle. The exact spatial location where ceramide is synthesized dictates its function. A ceramide molecule generated slowly in the bulk ER is a harmless intermediate destined for export. A ceramide molecule generated right here at the AAM is manufactured mere nanometers from the outer mitochondrial membrane. The cell intentionally places its most potent lipid weapon right next to the organelle it is most competent to kill. For years, cell biologists struggled to map the exact location of the presenilin proteins. They appeared to be scattered promiscuously everywhere across the cell. In 2009, a high-resolution analysis resolved the confusion. As we map these proteins onto our diagram, you can see that presenilin and its active complex, gamma secretase, descend directly into the MAM. Gamma secretase prefers highly ordered environments, making this cholesterol-rich raft its ideal address. Subsequent verification proved the entire amyloid processing line lives here. The precursor protein and beta secretase assemble side by side with the ceramide syntheses. The physical overlap of these molecules reorganizes the entire geometry of the disease. The genetic proteins whose mutations cause familial Alzheimer's share the exact same physical address as the cell's lethal lipid factory. To see where the failure begins, we have to look upstream of the final amyloid beta release, to the very first cleavage step performed by beta secretase. That initial cut leaves behind a 99 residue carboxy terminal fragment, simply called C99. In a healthy neuron, gamma secretase steps in and clears this C99 fragment almost immediately. As this animation shows, the early disease state breaks that clearance cycle. C99 is left uncleaved, and multiple fragments begin to accumulate directly within the MAM lipid raft. C99 does not sit idle. It behaves as an active cholesterol-sensing peptide. Its accumulation actively pulls extracellular cholesterol directly into the MAM. As cholesterol floods in, the contact structurally distorts. The raft widens, the organelle gap shrinks, and the bridging tethers multiply. The true early pathogen is the uncleaved substrate physically overworking the internal membrane, occurring long before the release of the extracellular plaque. The multiplied tethers lose their calibration, sending an unregulated flood of calcium directly into the mitochondrion. Simultaneously, the widened raft pushes sphingolipid turnover into extreme overdrive, rapidly overproducing ceramide. These excess molecules self-assemble, forming distinct physical pores that puncture right through the outer mitochondrial membrane. This altered outer membrane has a severe downstream effect on the inner workings of the mitochondrion. Deep inside the matrix, respiratory supercomplexes rely on a precise lipid environment to remain assembled. The sudden ceramide surge causes those structures to physically break apart. This disassembly crashes cellular respiration, starving the neuron of energy, while the outer pores leak lethal cytochrome C into the cytosol. The ceramide lipid disorder and the calcium bioenergetic failure share a single co-located executioner. Those disassembled supercomplexes also leak reactive oxygen species, sparking a devastating feed-forward loop. The resulting oxidative stress activates more sphingomyelinases, which drives further ceramide accumulation, which punches more pores into the membrane. If we map the certainty of these mechanisms onto a validity ledger, we can separate established fact from working hypothesis. The enrichment of the amyloid machinery at the MAM and the in vitro reality of ceramide channels are verified Tier 1 evidence. The claim that this dysfunction precedes plaques is an extrapolated Tier 3 hypothesis. We also have to explicitly discard dead ends. A widely cited 2003 paper claiming ceramide directly stabilizes the beta-secretase enzyme has been retracted. The mechanics of the tightened weld stand independently of that unsupported claim. Understanding the exact mechanical nature of this lesion dictates how we approach therapeutics, specifically the reflex to pharmacologically sever the AAM to stop the disease. Severing the weld would instantly kill the neuron. The cell requires this baseline lipid and calcium flux to survive. Future interventions must focus on tuning the set point of the tethers, or accelerating the clearance of the C99 fragment, rather than abolishing the organelle contact entirely. The plaques and tangles are the smoke. The buckling of this nanometer-scale weld is the hearth in which the long fire of Alzheimer's is laid.